Okay, so here we are, DNA lecture number 4. I'm going to also type up some notes and then I will upload those as well. So before we get into the actual PowerPoint, let me type in a couple of notes of definitions that I want to go through. Okay, so the first definition I want you to understand is a gene. And a gene is simply defined as, oops, let me take this off. Boom, boom. Okay, a gene is a linear stretch of DNA. So meaning the A's, G's, C's, and T's, okay, whatever order they happen to be in, that carries the instructions to code for a single protein. Okay, so that's what a gene is. And obviously, you know, lots of genes because your cells produce lots of different proteins. Okay, so the example I'm going to use in the lecture is the insulin gene. Okay, so insulin is a protein that helps control your blood sugar, and there is a certain stretch of DNA that corresponds to a gene for insulin production. So basically it's the instructions to allow your cells to make the insulin protein. Okay, second definition is gene expression. And gene expression simply refers to, once again, here we go, the turning on and off of genes. Okay, meaning that your cells are only going to express the genes that they need to. Okay, so you know you don't have to be making insulin all the time, so the insulin gene is not always going to be turned on. So genes are only turned on when they absolutely have to be. Oops, a little typo here. Okay, one more term I want you to understand is differential gene expression. Okay, and what that means is that the only genes that correspond to a certain cell type are going to be turned on. So it simply means that different genes are turned on in different cell types. Okay, so each one of your cells with a nucleus has the entire genome. Okay, so all 46 chromosomes, right? 23 pairs. However, the insulin gene is only turned on in cells of your pancreas. Okay, so brain-related genes are only going to be turned on in brain cells. Skin-related genes are only going to be turned on in your skin cells. And that's what differential gene expression means. Okay, so only genes that are needed in certain cells are turned on and off, even though we have the entire complement in all of our cells with a nucleus. Okay, and really the reason for gene expression is that, you know, expressing genes is actually a really expensive process in terms of energy, meaning to do it, you have to burn a ton of ATP, and cells are really pretty frugal. They're pretty cheap. So they're only going to express genes when they absolutely need to. Okay, so the processes I'm going to be talking about for gene expression are transcription and translation. So let's go back to the PowerPoint. Okay, so we had mentioned before about chromosomes, and chromosomes are just compact structures. It's a way to fit all of your DNA inside the nucleus of your cells. You know, all of the bacterial DNA inside a tiny little bacteria. But chromosomes are made up of genes, these linear stretches of DNA that actually code for protein. So they kind of have the appearance of like a bead on a string. So for example, if we look at a typical eukaryotic chromosome, a lot of time it has this banded appearance, okay? And if you were to look at, the DNA really closely, you'd see genes right next to each other, and they might have little spacer regions. There's a lot of your DNA that doesn't actually code for anything. But, certainly in the time that we're living in, more and more genes are going to be identified. So as of right now, over 30,000 genes have been identified in the human genome. That number is growing every day because lots of research is ongoing. And also, I just want to point out that, remember, the number of chromosomes and genes vary from species to species. Okay, so obviously you, as a human being, have different genes compared to a coconut or a bacteria or a squirrel. Okay, however, all of your DNA is made up of the same building blocks. It's just how much there are and what sequence they're in that makes us different from species to species. So when you think of what DNA does, DNA is really like the boss of a big company, okay? So think of a stereotypical CEO of a company where, here you have the big boss guy and he's just yelling instructions at his subordinates. So DNA is kind of like that, okay? The only purpose of DNA is to tell the cell when it needs to make proteins and then give the instructions so the cell can then make whatever protein it needs. So as a reminder, remember, proteins are the most numerous molecules in your cells and they do the most work. So it makes sense that DNA is going to carry the instructions to code for protein. So if your cell needs a lipid made, there's going to be a protein that makes it, okay? Usually an enzyme. If it needs a particular, hormone made, there's going to be an enzyme, which is a protein, to make that particular hormone. So yeah, proteins are doing the majority of the work, and they are by far the most numerous molecules in your cells, so it makes sense that DNA codes for proteins. So yeah, basically they do pretty much everything in the cell. So let's go back to the insulin example that I mentioned. Okay, so insulin is a protein. It helps control your blood sugar. Okay, this is actually the molecular structure of it. It's kind of a big fancy protein. So when the cells of your pancreas receive a signal that, hey, we're running kind of low on insulin, you guys need to make more, okay, it has to go through two different processes. Okay, the first is transcription. And what transcription does is it decodes the DNA, so reads the sequence of the DNA and transcribes it into a type of message molecule. Okay, and then that message molecule that is created through transcription travels to a separate part of the cell and is read and translated into a functional protein, okay? And they always happen in this order. Transcription always comes first, and then translation happens second, okay? So if there's no transcription, there's not going to be any translation, okay? This is why a lot of antibiotics act at the level of transcription in bacteria, because if they can knock out transcription, you're not going to have translation. Okay, so kind of makes sense. So transcription always has to come first before translation. Okay, so this is a slide I really like because it kind of shows how these processes happen. Okay, so first of all, let me zoom in on this. In my previous lecture video, we talked about replication. Okay, so let me emphasize again, replication has nothing to do with gene expression. Okay, all replication is it's making an exact copy of the DNA, so then the cell can reproduce itself. That's all it does. Okay, when we talk about gene expression, that's when we have the processes of transcription and then translation. Okay, so you have your original DNA molecule, the code is read through transcription, and it produces a molecule that is a messenger, and this is of a different type of nucleic acid called RNA, and specifically mRNA, which stands for messenger. Then the message travels to a different part of the cell and is translated, meaning that this code is then read and then translated into protein. Okay, so one of the things I want to make very, very clear, DNA doesn't turn into RNA, and then the RNA is not turned into protein. You simply read the code of the DNA to produce RNA, and then the RNA is read to produce protein. Okay, so, I want to make sure that that is clear. Okay, so first, let's talk about transcription. So the purpose of transcription is to read the code of DNA that corresponds to a specific gene, produce a message molecule, and then carry it in eukaryotic cells outside of the nucleus to a little protein factory that is called a ribosome. And bacteria also have ribosomes. Okay, so transcription and translation happen in bacteria and in eukaryotic cells exactly the same way. The only difference is that there's not a nucleus in bacteria. So to read the message, DNA is transcribed into another type of nucleic acid called RNA. And specifically, we call it mRNA or messenger RNA, which makes sense because it's carrying a message. Okay, so RNA is a tiny bit different than DNA. Okay, first of all, the sugar component in the backbone is a little different. It's ribose instead of deoxyribose, but also the base pairing rules are slightly different than DNA. Okay, and here is the main difference. There is no T in RNA. In RNA, instead of T, there is a base called uracil, which we abbreviate U. Okay, so for example, on a quiz or in an exam, if I show you a sequence of nucleic acid and I ask you, is this DNA or RNA? If there's T's in it, it's DNA. If there's U's in it, it's RNA. Okay, so in RNA, A always base pairs with U. Now, normally RNA is single-stranded in the cell, okay, meaning that you would only see like one strand of it. That's what messenger RNA looks like. I'm just showing you this double-stranded form so you can see the base pairing, okay? So instead of base pairing with T, A is base pairing with U. The C's and the G's are exactly the same. Okay, so let's look at the process of transcription. Okay, so remember, only the gene that needs to be turned on is going to be turned on. And it's only going to be turned on in the specific cell type. Okay, so in the example of insulin, only the insulin gene is going to be transcribed and only in cells of the pancreas. Okay, that's differential gene expression. Okay, so here's what happens. Every single gene has a start sequence, and that start sequence is a signal to the enzyme that carries out transcription, which is called RNA polymerase. Okay, remember, ASE means it's an enzyme, and hopefully the name tells you what it's doing. So in this sense, it's making a polymer of RNA. Okay, so at the start of the gene, the DNA unzips, which it can do, okay, because of its hydrogen bonds. RNA polymerase comes and sits right here, and then it starts working its way along the gene, performing transcription. So wherever it sees a T, it puts an A. Wherever it sees an A, it puts a U, and then the C's and G's, okay, and so on and so on. Also, every gene has an ending sequence, and this tells RNA polymerase, okay, we're done, you can fall off. Then what happens is the DNA winds back up, zips back up, and your messenger RNA, which is shown here in red, then floats out to a ribosome to be translated. Okay, so it's very gene specific. I mean, believe me, if all of your genes were turned on all of the time, you would literally explode. Okay, so, and plus you wouldn't be able to produce enough ATP for that to happen anyway. And actually, if genes get expressed in the wrong tissue, a lot of times we end up with a tumor. Okay, so for example, if the insulin gene keeps getting turned on in your brain tissue, you're probably going to end up with some type of abnormal growth because the insulin gene is not meant to be turned on in your brain tissue. It's meant to just be turned on in your pancreas tissue. Okay, so this is what transcription looks like. So this red strand, which is the messenger RNA, is going to float away and go out to a ribosome. Okay, so when the gene has been transcribed, so transcription is over, the messenger RNA detaches from the DNA. The DNA zips back up on itself and goes on taking its nap, okay? Then the messenger RNA is going to float over to a ribosome. And in the case of your cells, which have a nucleus, it's going to float out of these big holes in your nucleus called nuclear pores. Okay, so the messenger RNA then attaches to a little protein factory called a ribosome, and then we're going to begin the process of translation. Okay, so here you have your messenger RNA strand, and then these little guys are ribosomes. So ribosomes so let me put it this way. Okay, so floating around in the cytoplasm at all times, you have the two subunits of ribosomes. There's a small subunit and a large subunit. And actually, when they hook on to the messenger RNA, they kind of look like an upside down snowman. But you can see they actually have kind of weird shape. You know, they kind of look like mating whales. But they only come together to form an intact ribosome when there is messenger RNA present. Okay, so the messenger RNA carries kind of a magnet that tells ribosomes, hey, I'm ready for translation. You guys need to come and hook onto me. And remember, this is happening in bacteria as well as every type of eukaryotic cell. Okay, so now the ribosome has attached to the messenger RNA. And so here's the large subunit, here's the small subunit, here's your messenger RNA. Okay, so now we're beginning the process of translation. So to be able to read the code of messenger RNA and produce a functional protein, which are made up of amino acids, every 3 bases of the messenger RNA is going to be read at one time, okay? And every 3 base pair segment is what we refer to as a codon. Okay, you'll also notice this weird little molecule here that kind of looks like a rotten tooth. Okay, that is what we call transfer RNA. So it's a different type of RNA, and it's actually what links the codons on the messenger RNA to the amino acids which are being produced, to produce the protein that is being translated. Okay, so the ribosome then reads the code of messenger RNA And by using transfer RNA, it's able to construct the protein that the messenger RNA is carrying the code for. Okay, so transfer RNA is another type of RNA, and transfer RNA molecules float around in the cytoplasm all over the place. Okay, and each one of them is carrying a 3 base pair or codon segment, and we refer to this as an anticodon because it's going to base pair with the codon on messenger RNA, and it has the anticodon on one end, and on the other end, it has an amino acid. So what happens is in the center of the ribosome, you have a codon of messenger RNA, and basically all of these different transfer RNA molecules are coming into the ribosome, and whatever one properly base pairs with the codon on messenger RNA, the ribosome can recognize that and then keep that particular amino acid and hook it on to the growing protein chain. Okay, so the key is the codon present on messenger RNA base pairs with the anticodon present on the transfer RNA. And when these two base pair properly, the ribosome says, okay, we have a winner, and it keeps that amino acid and hooks it on to the growing protein chain. The transfer RNA then floats away, picks up another amino acid of the same type, and basically gets recycled. Okay, so transfer RNA is kind of an interesting little molecule. If we look at it in two-dimensional form, okay, at one end, it's going to have an anticodon. And so, for example, the anticodon shown here is GAA. So the codon that this would base pair to would be CUU on the messenger RNA. Okay, and it also has an amino acid at the other end. Transfer RNA is one of those rare cases where RNA is double-stranded, and it's because we want to keep recycling them, okay? We don't want them just to, be able to be degraded. And so they actually do have kind of weird bases in them. You'll notice some Ds, they have a psi, they have actually some Ts in here. And that's to make the RNA the transfer RNA sticky so it doesn't degrade. Okay, so getting back to what's happening, okay, this diagram again. So remember, the ribosome is reading the messenger RNA, 1 codon or 3 base pairs at a time. And you have all of these different transfer RNAs. There's actually 64 different transfer RNAs floating around in the cytoplasm. When the one makes it in there and it base pairs properly, codon to anticodon, the ribosome recognizes that. It keeps that particular amino acid and hooks it on to the growing chain, and then the transfer RNA floats out and is recycled. And so when you see ribosomes that are going through translation on a messenger RNA, they move along 3 base pairs at a time, and then the protein actually comes out the top of the ribosome. Okay, so the ribosome is sliding along the messenger RNA, 3 base pairs or 1 codon at a time. And when the protein is finished, when it reaches the end of the messenger RNA, the ribosome simply detaches, goes back to being its separate subunits. The messenger RNA is degraded, and then the nucleotides are recycled, and the protein floats off to wherever it's destined to be and, is able to be functional. So one of the things that's really handy is that it's not like one ribosome has to read the entire messenger RNA from start to finish before another ribosome can hook on. So you can actually have several ribosomes reading 1 messenger RNA at a time. So it works like an assembly line, okay? And this is it's a really cool system because it results in a lot of protein being made, Okay, so this is an electron micrograph picture, and you have to imagine that the messenger RNA is actually traveling, it's shown here, okay? And then these darker circles are ribosomes, and the little curly cues coming out the top, that is the protein being produced. But as soon as one ribosome is done reading a codon and it shifts to the next codon, then another ribosome can come and latch on to the messenger RNA. So the result is, from one messenger RNA, you can have a ton of different ribosomes attached to it at the same time. And as a result, you can produce a lot of protein really fast from one single messenger RNA. Okay, so we're going to stop there. I'm going to write up some notes for you, get those uploaded, and also upload some animation videos so you can understand these processes a little better. And then we'll come back and we'll talk about mutations. Okay, thank you.